PKU is an inborn error of metabolism. The clinical manifestations of elevated phenylalanine do not appear until 2 to 3 days after the birth. Phenylalanine is hydroxylated to form tyrosine by a mixed function oxidase, phenylalanine hydroxylase, which requires molecular oxygen and tetrahydrobiopterin, which is converted into dihydrobiopterin. If either PAH is deficient or tetrahydrobiopterin is not formed, this reaction will not take place and tyrosine will not form. Phenyl pyruvate, phenyl acetate, and phenyl lactate are not normally produced in significant amounts in presence of functional phenylalanine hydroxylase. However, these are elevated if there is a deficiency of phenylalanine hydroxylase or any of the enzymes required to synthesize or regenerate tetrahydrobiopterin and dihydrobiopterin. So here we can see that phenylalanine is elevated and it is diverted into the pathway for the formation of phenyl lactate and phenyl acetate. The normal concentration in young children and adults is less than 1 to 2 milligrams per deciliter. In newborn children, the upper limit of normal is around 4 milligrams per deciliter. In classic PKU, it may be greater than 20 milligrams per deciliter. So, phenylalanine concentration may be 10 times normal not only in plasma but also in the urine and body tissues of the patients with classic PKU. In classic or type 1 PKU, there is defect in PAH gene. DNA analysis has shown greater than 100 mutations including missense, nonsense, insertions, deletions in the PAH gene. Needle biopsy samples from liver of different patients with PKU and non-PKU hyperphenylalaninemia showed that PAH activity varied from less than 1% in patients with PKU to greater than 35% in patients with non-PKU hyperphenylalaninemia due to defect in the tetrahydrobiopterin production. As regards the diagnosis, prenatal diagnosis can be done through mutational analysis by DNA probes after amniocentesis or chorionic villus sampling. However, for postnatal workup, elevated phenylalanine levels may not be detectable until 3 to 4 days postpartum. Screening for PKU may be done through ferric chloride screening of urine or blood obtained from the heel prick can be used to perform a Guthrie bacterial inhibition assay using a special type of filter paper. In Guthrie bacterial inhibition assay, spores of organism Bacillus subtilis are plated on an agar plate containing an inhibitor of its growth. The blood obtained from infant in form of dried spot on filter dish is placed on the agar plate. If blood phenylalanine level is high or it is greater than 2 to 4, it will counteract the effect of the inhibitor of the bacterial growth and positive results will be obtained, and these results can be verified by HPLC. Apart from that, liver biopsy can be used to determine the level of the PAH activity. Patients with classic PKU appear normal at birth. If disease is not diagnosed and treated within the first month of life, infant develops varying degrees of intellectual disability and IQ scores less than 50. Delayed psychomotor maturation, tremors, seizures, eczema, and hyperactivity, along with stunted growth and dental problems. These are caused by the accumulation of phenylalanine 30 to 50 times the normal and due to production of phenylalanine metabolites like phenyl pyruvate, phenyl lactate, and phenyl acetate. all of which are associated with observed neurological and dental problems. Normal dopamine helps to keep the prolactin levels low. Since dopamine formation from tyrosine is compromised in PKU, as tyrosine is not formed from phenylalanine, this leads to high prolactin levels. The dietary treatment focuses on careful monitoring of phenylalanine intake to provide just enough for protein synthesis without causing phenylalanine accumulation. These preparations reduce the dietary intake of phenylalanine to 250 to 500 milligrams per day while maintaining normal intake of other dietary nutrients. Supplementation with tetrahydrobiopterin and hydroxyphenylalanine and 5-hydroxytryptophan may help, as tetrahydrobiopterin is the coenzyme, whereas hydroxyphenylalanine and tryptophan may help in neurotransmitter synthesis. Glycomacropeptide obtained from whey protein has a very low phenylalanine content and hence it is a useful supplement for PKU. Phenylketonuric individuals need to avoid processed foods and beverages containing aspartame. Aspartame is an artificial sweetener made up of aspartyl phenylalanine methyl ester because it is hydrolyzed to phenylalanine and aspartate in the intestine. Until gene therapy becomes available, the mainstay of treatment in classic PKU is phenylalanine restricted diet to maintain levels of phenylalanine in blood between 3 to 12 milligrams per dl. Although it is generally agreed that strict adherence to low phenylalanine diet is mandatory for first decade of life, less consensus exists regarding its indefinite use throughout the life, but there is an evidence that without observing lifelong dietary restriction, even adults will develop the neurological adverse effects of PKU. It is especially important for pregnant females to maintain phenylalanine levels during pregnancy. If women with PKU who are on phenylalanine restricted diet become pregnant, the fetus will not be harmed. But if the woman is not taking the phenylalanine restricted diet, the offspring can be affected with maternal PKU syndrome, as high blood phenylalanine has a teratogenic effect, causing microcephaly and congenital heart defects in the fetus. The neurological sequelae may result in part from the competitive interaction of phenylalanine with brain amino acid transport systems and inhibition of neurotransmitter synthesis. Large neutral amino acids such as tryptophan, tyrosine, histidine, and leucine share a transport system, the L system, with phenylalanine. Supplementation of diet with these amino acids can overcome high levels of phenylalanine in blood, allowing neurotransmitter precursors to enter the brain. As serotonin is synthesized from tryptophan, catecholamines from tyrosine, and histamine from histidine, if not corrected, these biochemical alterations lead to delayed myelin synthesis and delayed neuronal development. The individuals with PKU are fair colored because melanin formation occurs through tyrosine, but in this case, tyrosine is not phenylalanine. Here we can see that melanogenesis begins with conversion of amino acid tyrosine to dopaquinone. The addition of cysteine or glutathione to dopaquinone leads to the intermediates formation. followed by the subsequent transformations and polymerization to the final product, the phenylalanine. So here we can see that topaquinone is changed to the melanin pigment. Elevated phenylalanine crosses placenta. It is teratogenic for fetus, therefore it leads to microcephaly and heart defects. To summarize, we can see that the amino acid degradation pathway affected is the phenylalanine degradation. Missing enzyme in case of classic PKU or type 1 PKU is phenylalanine hydroxylase, whereas in case of non-classic PKUs or type 2, 3, 4 and 5, there is deficiency of dihydrobiopterin reductase or synthase. In both cases, there is mental retardation. Etiology is genetic and there is autosomal recessive inheritance, and both forms lead to intellectual disability if not treated at an early age.